Now we will discuss about some organelles and their functions. So these organelles are peroxisomes, proteasomes, and cytoskeleton. So these organelles are basically located within the cells and they perform various functions. So first we will discuss about the peroxisome. So peroxisome is basically membrane enclosed organelle and it is involved in specialized metabolic processes. Peroxisome is basically a membrane enclosed organelle and it is involved in various specialized metabolic processes. The main functions of peroxisome is one is beta oxidation, alpha oxidation, and synthesis. In the main function of peroxisome is beta oxidation of very long chain fatty acids or VLCFA. So it is strictly a peroxisomal process and alpha oxidation of branched chain fatty acid. It is another function of peroxisome. So it is also strictly a peroxisomal process. And another function of peroxisome is catabolism of amino acids and ethanol. It is also main function of peroxisome. So and peroxisome it synthesis bile acids and plasmalogens. That is important in phospholipid, especially in white matter of brain. So there are some disorders related to peroxisome. One is Zellweger syndrome, Refsum syndrome or Refsum disease, and Adrenoleukodystrophy. Zellweger syndrome it is autosomal recessive. It is caused due to defect or mutation in the PAX gene. Defective that results in defective peroxisome biogenesis and features of Zellweger syndrome are hypotonia, seizure, jaundice, craniofacial dysmorphia, hepatomegaly, and early death. These are features of Zellweger syndrome. Hypotonia, seizure, jaundice, craniofacial dysmorphia, hepatomegaly, and early death. These are some of features of Zellweger disease. And Refsum disease, it is also autosomal recessive. So when defect causes impaired alpha oxidation. So due to impaired alpha oxidation, buildup of phytanic acid takes place. So features of Refsum syndrome or Refsum disease is vision loss or retinitis pigmentosa, anosmia, hearing loss, ataxia, peripheral neuropathy, ichthyosis, cardiac conduction defects. So these are features of Refsum disease. So one is vision loss due to retinitis pigmentosa, anosmia, hearing loss, ataxia, peripheral neuropathy, ichthyosis, cardiac conduction defects. So treatment of Refsum disease is diet modification and plasmapheresis. So treatment is diet modification and plasmapheresis. So and another disease is adrenoleukodystrophy. Adrenoleukodystrophy it is X-linked recessive disease. So defect mutation in ABCD1 gene. Adrenoleukodystrophy. So adrenoleukodystrophy it starts with A, so we will remember by the the it so it it is also due to defect in the mutation in ABCD1 gene. So it also starts with ABCD1 and A for adrenoleukodystrophy. Then it causes defective beta oxidation. Due to defective beta oxidation, VLCFA buildup or very long chain fatty acid buildup takes place in the adrenal gland, brain, white matter, and testes. And features of adrenoleukodystrophy are progressive neurologic decline and adrenal crisis. And that these are some of features of adrenoleukodystrophy. Proteasome. This is another organelle, very important organelle that is located within the cell. So it is proteasome, that is basically barrel-shaped protein complex, and it degrades polyubiquitin tagged proteins. So some proteins are tagged with ubiquitin. So main function of proteasome, that is barrel-shaped protein complex, it degrades these polyubiquitin tagged proteins. So its main function of proteasome is maintain protein quality control. So it maintain quality of proteins. It plays roles in the immune response, basically MHC1 mediated antigen presentation. It is second function of proteasome. And when some defect happened in the proteasome, defect implicated in neurodegenerative diseases. So defects in the proteasome leads to neurodegenerative. Cytoskeletal elements, and these cytoskeletal elements, these are also located within the cell itself. So its definition is network of protein fibers in the cytoplasm. Cytoplasm is located where? It is located in the cell. So it is network of protein fibers in the cytoplasm. These cytoskeletal elements. The function of cytoskeletal elements is function of support cell structure. It cytoskeletal element, these support cell structure, movement of cell organelles. So movement of cell organelles within cell take place with the help of cytoskeletal element, and it also help in the cell division. So these are main functions of cytoskeletal elements. So types of filaments, there are three types of filaments: microfilaments, intermediate filaments, and microtubules. These are three types of filaments, and there are microfilaments, and their function is muscle contraction, cytokinesis, phagocytosis, actin. For examples of microfilaments are actin and microvilli, and intermediate filaments, they maintain cell structure. Examples of intermediate filaments are vimentin, desmin, cytokeratin, lamins, GFAP, for example, neurofilaments. Microtubules. The function of microtubule is movement and cell division. For example, cilia, flagella, mitotic spindle, axonal transport. For example, example of microtubule is centrioles. Microtubules structure. It is basically cylindrical outer structure. It is made up of alpha and beta tubulin heterodimers. Each dimer binds to GTP. It is incorporated into flagella, cilia, and mitotic spindles, and involved in slow exoplasmic transport in neurons. Negative end is near the nucleus, and positive end is toward the periphery. Motor proteins. The motor proteins are dynein and kinesin. Dynein is retrograde transport, positive to negative, and kinesin is anterograde transport, negative to positive. Clinical relevance. Clinical relevance is pathogenesis using dynein retrograde transport. So Clostridium tetani, poliovirus, rabies virus, and HSV. HSV reactivation, anterograde transport, kinesin mediated. Slow anterograde transport, rate limiting step in peripheral nerve regeneration. It is a rate limiting step in peripheral nerve regeneration. Now we will discuss about some drugs that acting on the microtubules. So these drugs are antihelminthic, antifungal, anti-gout, and anti-cancer drugs. These act on the microtubules within the cell. So mebendazole mebendazole is antihelminthic drug that act on the microtubule. Another is griseofulvin. Griseofulvin is antifungal. Colchicine. Colchicine basically is anti-gout drug that act on the microtubules. Vinca alkaloids. Vinca alkaloids are anti-cancer drugs that also act on the microtubules. Taxanes. Taxanes are basically anti-cancer drugs. These also work and act on the microtubules within the. So key takeaways from our lecture are peroxisome. We have discussed functions of different organelles within the cell. They have very important functions. These are functions of organelles like peroxisome, proteasomes, cytoskeleton, and microtubules. Peroxisome, they handle fatty acid oxidation and plasmalogen synthesis. And when defect in the peroxisome, defect causes severe metabolic diseases. When defect takes place in the peroxisome, they cause various metabolic diseases. Proteasomes. Proteasome basically degrade misfolded protein. Dysfunction linked to neurodegeneration. Dysfunction in proteasome, it is linked to neurodegeneration. Cytoskeleton. Cytoskeleton provides structure and movement. It includes microfilament, intermediate filaments, and microtubules. Cytoskeleton provides structure and movement, structure to cell itself and movement of organelles within the cell. And microtubules. Microtubules, these are essential for intracellular transport, cell division, and targeted by several drugs. So microtubules are very essential for intracellular transport, cell division, and some drugs we have discussed, they target these microtubules to work.